All right, let's jump right in. We're going to explore a question that, when you really think about it, is one of the biggest puzzles in biology. It's called the cancer paradox.
I mean, just think about it for a second. Your body is this incredible, bustling metropolis of trillions of cells, and they're dividing all the time. With that much activity, that many chances for a copy-paste error, you'd think things would go wrong constantly. So, why don't they? Why isn't cancer basically inevitable for all of us?
And here is the truly wild part. Get this. Research from Sinai Health has shown that the average healthy adult is walking around with millions of cells that already have cancer-causing mutations. The mistakes are already there. So what's the secret? What is the invisible force that's keeping these rogue cells in check? Well, that's the mystery we're cracking today.
To start our investigation, we've got to go inside the cell, down to the genetic level. And we're going to focus on two key types of genes that are, you could say, the prime suspects in this whole story. It's helpful to think of them as the accelerators and the brakes of a car. Now, this whole process of a normal cell going bad, it has a scientific name, carcinogenesis. It's basically the step-by-step journey a cell takes from being a team player to a renegade that just divides and divides without any control.
And that accelerator and brakes analogy? It maps perfectly to these two gene types. On one side, you've got oncogenes. They're the accelerators. When they mutate, it's like the gas pedal gets slammed to the floor and stuck there. All they do is shout, grow, grow, grow. Then you have the tumor suppressor genes. These are, you guessed it, the brakes. Their entire job is to slow things down or stop division altogether. But if they get broken, well, you lose all control.
The Pancreatic Cancer Action Network has a perfect way of describing this. They say when a gene like KRAS, that's a really common oncogene, mutates, the signal just gets stuck on go. It's a green light all day, every day, no stopping. Okay, so we know what's breaking, but how is it breaking? What's the inciting incident, so to speak, that causes this damage to these crucial genes in the first place?
Well, the threats are, frankly, everywhere. The damage can come from the outside, things like UV light from the sun or chemicals and tobacco smoke. It can come from certain infections. It can even come from inside our own bodies, from the natural byproducts of just being alive. And sometimes it's nothing more than a random typo, a simple mistake made when a cell is copying its own DNA.
But our bodies are smart. They have this amazing built-in safety feature, especially for those break genes. It's a concept called the two-hit hypothesis. See, you get one copy of each gene from each parent. And for these tumor suppressor genes, knocking out just one isn't enough. You have to take out the backup too.
So here's how it plays out. You get the first hit. Maybe you're born with it, or it happens randomly. But the cell is still okay, right? Because that second copy is still working, still applying the brakes. But then, if a second hit comes along and damages that last good copy, that's it. The brakes are completely gone, and that cell starts growing out of control. And once that cell has lost its brakes and its accelerator is stuck, the problem starts to escalate. We go from a single rogue cell to a whole neighborhood of them, a tumor. And from there, it can start thinking about spreading. That spread has a name you've probably heard, metastasis. This is when cancer goes from being a local problem to a systemic one. The cells break off, hop into the bloodstream or the lymph system, and basically try to set up new colonies in other parts of the body. It's what makes cancer so dangerous. But here's another twist in our mystery. Cancer isn't just one thing. It's not a single villain. It's more like a whole rogues gallery, and each one has a different personality, a different strategy, and a different speed.
Take a look at this, for example. These are different subtypes of breast cancer. A triple-negative tumor, on average, doubles in size every 103 days. That is incredibly aggressive. But an ER-positive tumor, it takes 241 days, more than twice as long. It's a completely different ballgame. And because every cancer is so different, modern medicine has had to become a kind of detective agency. To fight a tumor effectively, you can't just throw a standard treatment at it. You first have to investigate it, to crack its unique genetic code.
This is why that investigation is so important. Remember that KRAS gene we talked about, the stuck accelerator? Well, in pancreatic cancer, it's estimated that over 90% of tumors have that exact mutation. If you know that, you have a specific target to aim for.
And this is exactly what something called biomarker testing is all about. It's a pretty straightforward process, really. First, you collect a small sample of the tumor. Second, you analyze that sample in a lab to find its unique genetic fingerprint, its specific mutations.
And third, doctors use that info to decide on a personalized treatment plan. It's like instead of a master key, you're finding the one specific key that fits that one specific lock.
So we've looked at the broken parts, how they get broken, and how we investigate the crime scene. But there's a final clue from some really recent research that might just blow this whole case wide open. And it turns out it might all be about timing.
Okay, here is the bombshell finding from that Sinai Health study. What if the mutation itself isn't the most important part of the story? What if the real deciding factor is the speed at which the cell with that mutation is dividing?
The evidence they found is, honestly, mind-blowing. In every single case they looked at, the cancer came from the cell type with the fastest division time. The speed demons were always the ones to turn cancerous. But here's the craziest part. When they found a way to artificially slow those fast-dividing cells down, the tumors just didn't form, even when the mutation was still there.
This changes everything. It suggests that our whole mental model of cancer might be incomplete. We've been so focused on the genetic typo, the broken part, when maybe we should have been looking at the pace, the rhythm of the cell itself.
And this really bumps up against the way we usually talk and think about this disease. For decades, the main metaphor has been war. We fight cancer. We battle tumors. A study on this found that people very often visualize cancer cells as literal monsters that have to be attacked and killed.
But what if this new research is pointing us toward a totally different way of thinking? What if the key to solving the cancer paradox isn't just about building bigger weapons to slay monsters? What if it's about something more subtle? About understanding the environment of our cells and learning how to gently tap the brakes, how to just slow things down enough to let our body's amazing natural defenses do their job? Now that's a question worth thinking about.